Let's start by talking about Bacillus bacteria. These are rod-shaped bacteria that often form chains or tangled groups. They're pretty unique because of their shape and how they group together. Understanding their basic form helps us recognize them under the microscope and sets the stage for learning about their behavior and impact. Now, Bacillus bacteria can produce spores, which are like their survival capsules. Spores are produced by gram-positive bacilli. Some of them are aerobic, meaning they need oxygen, and they have non-bulging spores. Others are anaerobic and can have either bulging or non-bulging spores. This distinction is important because it affects how they survive and cause disease. Bacillus bacteria have several key traits. They're gram-positive rods, which means they stain purple and have a thick cell wall. They can be aerobic or facultatively anaerobic. They form heat-resistant endospores that help them survive harsh conditions. Most are motile with flagella, except Bacillus anthracis, which isn't. They also produce catalase and ferment glucose, creating acid. These features help us identify and understand their biology. Let's explore where Bacillus bacteria live and what they do. They're everywhere, in soil, water, and even on culture media, where they can contaminate samples. Some species are saprophytes, meaning they help break down organic material. Two medically important species are Bacillus anthracis, which causes anthrax, a disease that can jump from animals to humans, and Bacillus cereus, known for causing food poisoning and infections. Focusing on Bacillus anthracis, it's one of the largest bacteria that cause disease. It has a rectangular shape and can appear singly or in pairs, sometimes looking like bamboo sticks in culture. Its spores are oval and central, but don't bulge and form only outside infected tissue. It has a polypeptide capsule visible with special stains, and unlike others, it's non-motile. It stains gram-positive and shows specific reactions helpful for diagnosis. The virulence of Bacillus anthracis depends on several factors. It produces heat-stable spores and a protective capsule that help it evade the immune system. Its anthrax toxin has three parts: protective antigen, which helps the toxin enter cells; edema factor, causing swelling; and lethal factor, which damages cells. Together, these make the bacteria particularly dangerous. When it comes to how anthrax develops in humans, exposure can be industrial or non-industrial. Industrial cases occur in workers like those in meat packing or wool sorting. Non-industrial cases happen in occupations such as veterinarians, butchers, and farmers who have contact with infected animals. Understanding these exposure routes helps in prevention and diagnosis. Anthrax itself is a zoonotic disease, meaning it passes from animals to humans. Animals usually get infected by ingesting spores from soil. Direct animal-to-animal spread is rare. The disease often leads to fatal septicemia, but can sometimes be localized. Humans get anthrax either directly or indirectly from animals, highlighting the importance of controlling infection in animal populations. Cutaneous anthrax, also known as Hyde-Porter's disease, starts when spores enter the skin. It begins with a small bump, often on exposed areas like the face or hands. This bump turns into a fluid-filled blister called a malignant pustule. Sometimes the lesion heals on its own, but if untreated, it can lead to fatal septicemia or meningitis in up to 20% of cases. This progression shows why early recognition and treatment are critical. Pulmonary anthrax, or wool sorter's disease, primarily affects wool factory workers. It's characterized by hemorrhagic bronchopneumonia, which is a severe lung infection with bleeding, and it has a high fatality rate. Sometimes it can lead to hemorrhagic meningitis, a serious complication. This form underscores the occupational risks associated with anthrax. Intestinal anthrax is less common and usually happens in communities that consume animals that died from anthrax. It causes violent inflammation of the intestines, leading to bloody diarrhea, and has a high fatality rate. This highlights the dangers of ingesting contaminated meat and the importance of safe food practices. Here, we see images related to anthrax. One shows workers handling wool, which is relevant because this is a common source of exposure in pulmonary anthrax. The other image shows a large black lesion on human skin, a hallmark of cutaneous anthrax. These visuals help connect the clinical and occupational aspects of the disease. Bacillus cereus is another important pathogen. It causes gastrointestinal infections and infections related to catheters. It has two main forms of food poisoning: the emetic form, often from contaminated rice, causing vomiting and cramps, and the diarrheal form, from meat or vegetables, causing diarrhea and cramps. Both forms involve heat-stable enterotoxins, making them resilient to cooking. Bacillus cereus differs from Bacillus anthracis in several ways. It's generally motile, unlike the non-motile anthasis. It lacks a capsule, grows in short chains rather than long ones, and doesn't produce the characteristic medusa head colonies on nutrient agar. It also shows strong hemolysis on blood agar, whereas anthracis shows little or none. These differences help us distinguish between the two in the lab. Diagnosing bacillus infections involves collecting specimens like swabs, pus, sputum, or blood. We use gram staining and special stains like McFadden's reaction to identify the bacteria. Culturing on nutrient agar reveals the distinctive medusa head colonies, and other tests like gelatin stab and Ascoli's thermoprecipitation help confirm the diagnosis. PCR can also be used, especially in bioterrorism cases. Treatment typically includes antibiotics like doxycycline and ciprofloxacin. However, these drugs don't neutralize the toxins produced by the bacteria, so supportive care is also important. Preventing infection involves good hygiene and sterilization practices, especially in industries handling animal products. Early treatment improves outcomes significantly. Prophylaxis focuses on preventing anthrax by improving hygiene in factories and properly sterilizing animal products such as hides and wool. Animals suspected of dying from anthrax should be buried deeply with quicklime or cremated to avoid contaminating soil. Vaccinating cattle with the Stern vaccine is also a key preventative measure, helping to break the cycle of infection. Finally, it's important to note that aerobic spore-bearing bacilli, like anthracoid bacilli, are common contaminants in labs. Some species, like Bacillus thuringiensis, have biotechnological uses, and Bacillus stereothermophilus is used to monitor autoclave sterilization. Also, some other zoonotic diseases that can be caused by different bacteria, reminding us of the broad impact of infectious agents in both animals and humans.